Evening all. Okay, so year 13, I'm going into a bit more depth in terms of inheritance today. Yesterday we covered some of the more simple stuff, the monohybrid, dihybrid crosses without any complications, but effectively we've got a bit more trickiness, and I've done meiosis already in the year 12 stuff, so if you've got a solid understanding of meiosis, this is going to be a breeze. But I'm actually just trying to find where we are on YouTube right now so I can see the comments. Why are you not showing me as live, YouTube? So what is it that you find difficult about this topic? It's universally found super difficult in general, and I want to make sure that, do you understand why you find it difficult or not? Okay, sweet. So in which case, let me just, okay, sweet, which has done it for me. Inheritance, we're doing a bit more complex inheritance is what we're doing today. Again, I'm just going to share my screen for those of you that are new and joining us. The Study at Home Exambulance is the course that we've put together to give you some structure and to support you whilst you're studying at home. And yeah, let me just show you how to do that quickly, because we are going to be quite tight on time because this is a tricky topic and I want to try and get some questions in as well if we can. So let me actually just choose the right window first of all and the right program to share with you. So just from our homepage, tailoredtutors.co.uk, you will see the Study at Home Exambulance. We're probably going to build an entire page for this. Everything's super rushed and just thrown together. We just wanted to get you in there as quickly as possible. So obviously you've got the biology, the chemistry and the maths. So if you want to sign up to that, then that's totally fine. We've given you a study schedule for the 3 different phases, finishing the content first strategy, and then keeping basically your exam technique up to scratch, practicing some of the revision, TT revision method. And then if you're going to do the exam, if you don't like your calculated grade, then you've got your means of redress. You can do the exam, which is probably September. It says autumn, but we're thinking September. We don't know that for sure. So this is the proposed strategy. I'm actually I'm not around on Friday because I'm going to be on a plane again. So just let you know that I'm not here Friday, but I will be here tomorrow. Same time, same place. Okay, epistasis is not, I'm not going to go into epistasis today because it's not done by all the exam boards. I'm going to make sure that this is applicable to everyone. So we've got OCR, AQA, EdXL. It's going to be relevant to IAL. It's going to be relevant to the WJ, cover all these things that I'm going to be doing today as well. Should you start with past papers on before 2015? Can you just rephrase that so I can understand the question and I can answer your question. Yeah, who is it that does epistasis? Epistasis is covered, epistasis and epigenetics. OCR don't do epigenetics and EdXL don't do epistasis. So I'm pretty sure I'm the right way around with that. Okay, let us go into, yeah, let's share my screen, but I need to switch apps first. So autosomal linkage. Autosomal linkage, a tricky one. Again, it's understanding what's happening to the chromosomes to basically let all this happen, to what's controlling how this all works effectively. Let me just make sure I've got all the chat visible. Let's put it into a new window. Something like that. Yeah, I'd say that works pretty well. Okay, let's choose a pen. I'm going to go red, but straight in with the key term. As usual, what is an autosome? An autosome is any of the chromosomes that are not the sex chromosomes. So it's all the non-sex chromosomes, basically. Oh, can't even write the word sex. Sex. Chromosomes. Oh dear, that is shocking. Let's write this properly. Chromosomes. So how many does the human have? So their chromosomes numbers 1 to 22, but chromosome 1 is both homologous pairs. So there are 44 autosomes, and there would be 22 homologous pairs. But you're right, they're numbered 1 to 22. But chromosome 12, there's 2 chromosome 12s. One's from your mum, one's from your dad. They carry the same genes, but they have different alleles. We covered this already. So basically, what means something is linked? Whether it's sex linked, whether it's autosomally linked. Genes are linked. Genes are linked if they are, now that I've written, and just as I finish writing it, one pops into the chat. If they are on, or maybe have loci on the same chromosome. So it's a pretty simple concept if they're on the same chromosome. Obviously, that could be a sex chromosome if they're, but the sex chromosomes behave a bit differently because the Y chromosome is so much smaller than the X chromosome. We'll come on to sex linkage afterwards. What does this mean? This means that they will stay together, or yeah, they will stay together during another key term. They won't be separated, in other words, during independent assortment or segregation. Which in other words means they will be inherited together. They don't have the same loci or loci, because if we look at my, I haven't drawn them yet. That's why you haven't seen them yet. Let's draw some examples here. So we've got, I'm going to draw a homologous pair. How much space can I steal from in this? I'm going to draw its homologous partner. I'm going to cheat and just make my pen fat for the solid one. I generally use like solid and not solid for my homologous pairs, just to differentiate, but the same color because they contain the same genes, but different alleles. So during independent assortment is when they line up, segregation is when they separate. We're going to basically do this, and this one is going to go in this direction. This one is going to go in this direction. So let me draw some alleles on here. So let's say this organism is heterozygous for T and R. I think we use T and R from our examples that we've used so far. I'll try and be consistent. So it's big T, little t. I've used red for my R's, so I can put that in red, I suppose. And then big R, little r. But because they're on the same chromosome, things, well, we're going to do the locus for T is going to be here. Obviously, the locus is going to be the same. We know that these sister chromatids, there's going to be the same allele either side. So one of them is big T, one of them is little t. And then we have, let's say, our R allele locus is down here. We've got one is going to be big R, one is going to be little r. But this is something I showed yesterday, and I think a better way of showing you this is in this over here. So we've got various scenarios here. Is it going to let me just move all of this? Oh, it might let me move everything. Something like that. Oh, maybe not. I think it's maybe more trouble than it's worth. Okay, so this is what I've drawn over here. We've got linked our T and our R. See, all the genotypes here are big T, little t, big R, little r. The difference is when they're not linked. So I've drawn my big T, little t, big T, little t. So these are the same. But if they're not on the same chromosome, then you've got a different pair of chromosomes and the R's are down here. And so there's two possible arrangements if they're not linked. You can have different arrangements. So you can have the big T with the big R here, and you can have the little t with the little r here. So assuming no crossing over takes place, we're going to get the alleles big T, big R, and we're going to get the alleles little t, big R. But it's random independent assortment as to which alleles end up on, or which of the homologous pairs end up on either side. So you could have the T's are lined up the same way here, but I've just flipped over and just said that the other chromosome just lines up the other way around. And so that's also totally possible and equally likely, equiprobable. So you could also have the big T, these are the first meiotic division is gonna divide down here. Again, these are gonna go this way. And this would result in big T, little r, and you'd have little t, big R. So the four possible alleles you can get from an organism that is big T, little t, big R, little r, and if they're on different chromosomes, where you've got equal numbers, equal chance of getting any of these. So there's 25% chance of any one of those, your sperm containing, or eggs containing any one of those. Whereas if they are linked, it is only possible when these divide, unless crossing over separates them, which I'm not going to go into that just at this moment in time, you're always going to get the big T with the big R, and you're always going to get the little t with a little r. You can get some other recombinants through crossing over, but because they're not on different chromosomes and you can't shuffle them, there's only one way they can line up during independent assortment. And therefore, the only outcomes you can get, the genotype would look the same, but these are linked. These are not linked. And so the only possible alleles you would get from here are this or this. Obviously, if you're just given this in the exam, you don't actually know, does the big T go with the big R or does the big T go with the little r? But if you're ever left in any doubt, there was one WJ paper once that didn't make that distinction. Catastrophically bad exam paper writing. They should tell you, you would assume that the dominants go together and the recessives go together, just because the way that breeding experiments work, you would take the purebred dominant cross with the purebred recessive, homozygous dominant, homozygous recessive to produce heterozygotes, and you'd more than likely have that scenario. Okay. Yeah, because of independent assortment, these are alternative scenarios. It's one or the other. This is here. Whereas, you're just going to show me the screen. Okay, perfect. If they are linked, they will be inherited together. So we can see that basically the big T and the big R are going to go together, and the little t and the little r are going to stay together. The only way that you can shuffle them up is during, my iPad is doing some interesting things. If you get some crossing over taking place and you get, let's say, this one coming over here and this one over here, and then we get a little bit, we can swap over, and crossing over can give you some of the other alleles. So the main alleles that we're going to get, by number, the vast proportion are going to be the same as the parents. They're going to be TR or little t, little r. We'll get a few, if crossing over takes place here, we could get a few of these, and we could get a few of these, but their crossing over doesn't happen every single time. And so it's not, well, it certainly doesn't happen between those two specific gene loci every single time. So we have many of these and many of these, and we have few of these. So it's much more likely that you have the same, again, the same genotype as the parent. So yeah, if they're on the same chromosome, independent assortment can't separate them. And so they are inherited together. These are the gametes that can be made by the parent who is like this. Okay, so where do we want to go from here? I'm going to say that autosomal linkage, I might put that in red because it's key term. This is going to mean that you have, what's going to be the result of the offspring? It's going to mean that the offspring will have the same, it's a key term, it should go in red, I can't say, genotype and phenotype. So the same genetic makeup and the same phenotype as their parents. Because they're going to inherit, because the shuffling doesn't really take place. Matilda. Okay, so I see you're talking about the 93 to 3 to 1. You, in a normal dihybrid cross, if you were just given this and you say do the dihybrid cross, you would assume in normal world that they're not linked. You would assume that this is going on. Therefore, you'll have equal percentages or proportions of each of the 4 possible gametes. And then if we do our cross like this, and we were to draw this out, you would get a 9 to 3 to 3 to 1 ratio if it's not linked. I'm going to put a big fat line down there. However, as we've ascertained, basically, if they are linked, independent assortment can't mean that you have 50-50 random chance which T goes with which R, because this T is always going to stay with this R unless crossing over takes place. So now let's just maybe a good chance to just have a little look at that. So we've got our big T here, our little t here, and our big R here, and our big R here, our little r here, and our little r here. So after this cross, let's get rid of this. Well, at the end of meiosis, we're going to have these basically. This one's going to be unaffected, big T. This one's going to have the big T, this is little t, and this is little t. This one's going to have the big R, so that's these two. This will have done its crossover here. So this one's going to have the little r. This one's going to have the big R. Again, we're going to have we're going to have many of these. We're going to have many of these. These are the gametes, the genotype of the gametes. And they're going to have few of these ones because these only result from crossing over, and crossing over doesn't happen most of the time. Only made by crossing over. So if crossing over takes place, it's possible to separate them and to produce some of these gametes, but they're not, it's not equal numbers. Here we've got equal numbers, and therefore we end up basically multiplying them out equally. You've got equal numbers and you get the 9 to 3 to 3 to 1, but that assumes you have equal numbers of big T, big R, little t, little r, big T, little r. You have equal numbers of each, but we don't have that scenario if they're linked. We have basically, they're nearly all, you're probably going to have something like, I don't know, 45% this, 45% this, and then what have we got left over? We're at 90, so then 5% might be have this and 5% like this. So it's nowhere near equal. So if we actually do our cross, basically nearly all of them are going to come out as the parents were. So we're going to get most of them. Going to ratios, it would be more, it's basically going to behave like they're one gene because they are a one chromosome. They're more likely to be inherited a bit more like a monohybrid because effectively they stay together. The vast majority of them are inherited together. So it'd be more, it'd be more similar to monohybrid inheritance because they're basically just inherited together the vast majority of the time. Crossing over is an exception that occasionally crops up to separate some of the alleles to make a few rare gametes which have a different genotype. And obviously, if depends on the loci or loci of the let's draw both of them. If the same alleles here, if we have no, too fat. If we have T's here, I'll draw the T's in the same place on both of them. And we have our R's as I've drawn them down here. Or if our R's are up here, which ones are more likely to be separated by crossing over? I'll give you A and B. Which ones are more likely to be separated by crossing over? The ones that are further apart. Okay, so these are more likely to be separated by crossing over. I mean, like if you were going to if you're going to randomly cut with a pair of scissors across here somewhere, with, if you were just to take a random snip point here, like you're unlikely to get that line directly in the middle, right? It's basically, I can't even draw a line between the two over here. You're unlikely, it wouldn't be across both sides, it's maybe misleading. It would be like this. It's unlikely to happen there, right? Because they're right next to each other. Whereas you've got a long distance between them, it's relatively, and sometimes they do super weird crossings over where you get like, does that kind of make sense that it crosses over between here and here and they just swap that middle section? And it's not always the ends, can happen in many different ways. We don't go into huge amounts of detail in the A-level biology spec. The thing you need to know is that the closer two genes are on the same chromosome, the less likely it is for them to be separated by crossing over, or alternatively, the further apart they are on the same chromosome, the more likely they are to be separated by crossing over. So if I'm going to put this on my actual template note here, there was a big point here that I've decided to not include. So that space. So is okay? This is complex. Is everyone up? Is everyone with me? Have I lost anyone at this point? Because this is one of the most complicated things to get your head around in A-level biology. So maybe I could say the same genotype of their parents, which was big T, big R, little t, little r. So the genotype was this. Why will the offspring of parents that have autosomally linked genes have the same genotype? It's because you've got to get your head around the fact that the alleles, the alleles that are passed on into the gametes basically you can just assume that all of the alleles are going to be the same as their parents. Just that's the easiest. Forget the crossing over takes place. Just assume that all the alleles are either big T, big R and little t, little r. So if we cross, maybe if we do that as a cross, we have this, and then we end up with the same as we had before. So it's all about, because they can't be separated at independent assortment, they make basically this, they can't be separated. And that means that they produce unequal, the gametes that they produce are more likely to produce offspring that are the same genotype and the same phenotype as their parents. Unless the alleles are separated during crossing over. Are you gonna let me just draw fat chromosomes? It's gonna be way quicker if I can just draw. Them like this. And we've got, what have I done here? I've done big T's at the top, little t, little t. I'm not going to add an extra allele just to make it a bit. No, actually, it's probably a bit further down. The T's are not affected in this particular example. And if I were to add in another allele, which I might want to do in purple, and have it as Q, and have, let's forget allele Q. We don't need to know. I explained that on the other page, that the closer two alleles are together, the more likely they are to stay together, or the further they are apart, the more likely they are to be separated. So let's put a sentence on the page about that. So the closer two the closer the loci of two genes, we could say the more closely linked they are. And again, we can say the majority of the offspring will have the same genotype and phenotype as the parents. I'm really struggling to write, man. Because in these breeding experiments, you basically always inbreed the offspring. Maybe that's where I wasn't making myself totally clear, that once we've produced these gametes here, we've produced many of these and many of these with relatively few of these, you are then going to inbreed, so take the products of this, these gametes, and you're going to inbreed them with themselves. That's how we get the 9 to 3 to 3 to 1, is that we take, I wonder if I've still got it here. No, I think I'd removed them from my PDF viewer, but that's how we get the 9 to 3 to 3 to 1. We go with homozygous dominant parent, homozygous recessive parent, breed them together to produce the first filial generation, the F1 generation, 100% homozygous. Then we inbreed the F1 generation with themselves, so take the offspring and breed them with themselves to produce the F2 generation, which is when we see all the quirky stuff pop out. And that's when we get the 93 to 3:1 if they're not autosomally linked. So if they are autosomally linked, typically you take the same process, you do the same thing, and this is basically would be the F1 generation, and then the products of this would be the F2 generation. It would be more similar to a 3:1, but it's not going to be nothing's ever an exact 3:1 because of random fertilization. You don't know which ones on average you get 3:1 with monohybrid inheritance. Same with on average you get 93 to 3:1 with dihybrid inheritance. It would be more similar because it's basically behaving as one gene because they're inherited together pretty much always, but not always because of crossing over can split the odd ones up. Okay, how are we doing for time? 15 minutes. Okay, we've got a choice, and I give you the choice. You are the people. We can do sex linkage in terms of theory, or we can do question on autosomal linkage. If the genes are autosomally linked, you do not get a 93 to 3:1. Everyone wants to do questions, basically. Okay, so I'm just going to go back to the question. Yeah, if they are autosomally linked, you do not get the 93 to 3:1 ratio. It would be more similar to a 3:1. But I wouldn't give ratios there particularly. A common question is like the 93 to 3:1 ratio has not been observed in these data, suggest a reason why. Now it could be that you've got the genotypes and phenotypes are more similar to the parents. You have most of the same genotypes of the parents. Let's find a question. I think that's in this here device. It must be in here because I put it in here. Oh no, it's not in here. It's in the other place. Okay, let's have a look at this. Let's share my screen once again, and we'll see if we can tease out both of these just quickly. Does crossing over happen for non-linked? Well, if they're not linked, they're not on the same chromosome. So you can't, crossing over, it only exchanges material between homologous pairs. So if they're not on the same homologous pair, you can't get crossing over between them. Does that make sense? Okay, genes are for fruit, genes, okay, in fruit flies, genes for, blue is my question, ask this color, body color and wing length are linked. What does this mean? I'm going to leave you to it. Super easy, that one. We've covered that already today. Okay, the scientist investigated linkage between the genes. So what do we know that they're on the same chromosome? Okay, done. So that's what's going off in my head at this point. Genes for body color and wing length. He carried out crosses between fruit flies with gray bodies and long wings and fruit flies with black bodies and short wings. Okay, so that's what we've got. Big G, dominant allele for gray body. Little g, black body. Big N, dominant, long wings. Little n, recessive, short wings. Gives us homozygous dominant, homozygous recessive, 100% heterozygous in the offspring. Okay, they're giving us, this is the F1 generation, not that we really need to know it by that name. These offspring were crossed. So, okay, so here we need to pay attention. These offspring were crossed with flies homozygous for black body and short wings. Well, what gives us black body? Black body is this one. Black body homozygous, must be little g, little d. Homozygous for short wings is little n. So this is the genotype of this organism here. Okay, so we're crossing these offspring. This is our cross. Everyone aware of how I've got here. Translating the words into the genotypes is difficult here, not always obvious. Okay, so that's what I've got. So again, in fact, they've actually given it to us here. We didn't have to do that. So number of offspring. What were our parents? Our parents were, okay, we don't actually have, okay, all gray body with long wings. They've given us the phenotype here. So we've got gray body, long wings. Yeah, and then we've got black body, short wings. Black body, short wings. So now, like Nino, we've got major red, well, not red flags, massive flags going on here. And we've got lots of these, right? And we've got relatively few of the others, the other two possible outcomes. So use your knowledge of gene linkage to give reasons or to explain these results. And this is a 4-marker I give my students. This is one of my handouts from my live class. Very little space because I'm training you to be concise with your wording. This is one of the main skills in A-level biology, getting to the point quickly, understanding what the question wants. And so I don't give my students lots of space because I'm forcing you to pare down the information to what you need to know to stop you waffling, to save you time, so you've got more time to think. Basically, if we decode this question, we get explain autosomal linkage, more or less. Like it's like we can go back to the first principles here and explain it relatively from kind of explain. Well, how would I rephrase this question? It's more simple than you think. I would say that the genes are linked. They're on the same chromosome. Therefore, they produce more gametes with the genotype, with the parent's genotype. Okay, so that basically explains why we have these for 2 marks. And then we need to explain why we have these for 2 marks. I'd say in my head, that's how I'm answering this question. We have lots of these because they're linked and you produce more gametes. We could even name the gametes. The gametes would be probably big G, big N, and little g, little n. Yeah, which you can only produce little g, little n in the other one. I've lost the chat. I don't know whether I've lost you in general or not. Okay, I'll show you the mark scheme. Let's see in a bit here. You're going to be a bit lost otherwise. Okay, in fact, I'm going to show you the mark scheme. This question isn't finished yet, and you're going to see the answer otherwise. So, okay. If the genes were not linked, what ratio of phenotypes? Okay, so let's just go back to what we had here, because I distracted you quite a lot here. These were our parents. Okay. The question is, so if I'm going to rephrase this question to make it easier for you, some of you may have, if they were not linked, what would you expect? Now, a lot of you jumping in with 93 to 3 to 1, and I try not to predict these too much. Okay, so let's just do the very simple cross. You have big G, big N, little g, little n, little g. We're actually going to have, if they're not, okay, so if they're not linked, actually, I'm making a mistake here. So we've got this. If they're not linked, then all combinations are possible and equally possible. and then we're going to cross that with little g, little n. That's the only gamete that the homozygous recessive can make. So what do we get if we cross this? Okay, so we've got one big G and one big N. So this is, I think, long and gray. And then we've got long wings and black. Then we've got short wings and gray. I think I'm getting this the right way around. Maybe I'm getting it the wrong way around. And then we've got short wings and black. One of these, one of these, one of these, and one of these. I think actually I've got my genotypes, my phenotypes the wrong way around, because what were my genes? This was gray and this was black. So I have done them wrong. And then this was, I think, long and this was short. So let me just rewrite them. But the answer is 1 to 1 to 1 to 1. Because you're crossing it with a homozygous recessive, and this one has basically one of each. That's how it ends up that way. So this would actually be gray, long, gray, short, black, long, and black, short. What statistical test would you use? Inheritance, basically, which statistical test do you always use with this kind of stuff? Why is it not 93 to 3 to 1? I've literally just drawn it for you, why it's not 93 to 3 to 1. Because you're not crossing it with itself. It would be 9 to 3 to 3 to 1 if you were doing this cross. With itself. So that would be with itself. You can't just use this cross would give us the 9 to 3 to 3 to 1. But we're not crossing it with itself. We're crossing it with homozygous recessive. So it's everything is going to be compared to a recessive gene, which means all of these will be expressed because you're giving it, putting it up against a recessive. If it's dominant, it's gonna just override the recessive, and you're going to see the big G. If it's recessive, then you're going to be homozygous recessive. You're going to see the little G. Again, you basically, because you're comparing it to a recessive, you're going to see everything. And this has got 4 different alleles, so you're going to get the 4 different traits playing out. So you can't just jump in with predicted the phenotypes or the phenotypic ratios. You basically have to follow the question through, and the new spec is going to test you like that. That's how it's going to be. And yeah, it is the chi-squared test. And give a reason. Anyone who was in my statistics live class the other week, what's the reason for chi-squared test? That's what you would think to say observed and expected. Categorical data is what this mark scheme is going to say, because this is actually an AQA question, and that is the AQA wording, but it's looking at categoric data. So the data fits into categories. It is this phenotype or that phenotype. So there's discrete different categories. So you have gray body, long wings, gray body, short wings, black body, long wings, black body, short wings. It's a category. And therefore, because it's categoric data, that's generally what the mark schemes are going to say. If you've got to justify students' t-test, you can say that you're comparing for the difference between 2 means. Or if it's correlation coefficient, you can say you're looking for a relationship or a correlation between 2 variables. Ooh. Okay, so I want to know on a scale of 1 to 10 in terms of how much you have that has helped with your understanding of inheritance. This basically all comes back to meiosis and what those chromosomes are doing, being able to visualize, oh, well, if they're on separate chromosomes, then you can arrange them in different combinations because independent assortment is random. Whereas if they're on the same chromosome, you can't shuffle it. Best thing to do right now would be to go and do autosomal linkage questions to kind of test yourself on all of this. And if you get stuck, try drawing out the chromosomes. Try seeing what happens to those chromosomes and try following them through. And that really kind of goes back to meiosis, which is why I started underpinning all of this with meiosis in the AS live class. If you missed it, the recording is on YouTube. We're just putting our playlist together, but it's definitely search the Taylor Tutors channel. It was the live class, AS live class, numero uno. Ruhab, where can you find questions? The exam guide. Straight in the exam guide, under the inheritance section, autosomal linkage questions. The WJEC, download all the WJEC past papers, open them all and search for autosome or search for linkage. If it's linked, generally, you're going to get some crossing over, but not a huge amount. And that's where you get those other gametes being formed. And those other gametes are the ones that when you inbreed, you get a few of the genotypes and phenotypes that were not the same as the parents. It's a tricky one to teach. It's a really, because you basically need your understanding of meiosis to be rock solid. Because when I'm talking about independent, so people asking me in the chat, what is independent segregation? If you don't know that, go back and get that hammered out first, because trying to do linkage without understanding meiosis is like trying to juggle 5 balls before you can juggle 3 balls. Like there's a progression. You need to understand the preceding steps before you can really understand it. Otherwise you'll just end up learning ratios like 93 to 3 to 1. You'll see, oh, what is the ratio? I've got 2 alleles, I've got 2 genes, must be 93 3 to 1. You're just trying to like remember answers rather than understanding the principles. Like I'm all for taking shortcuts when it can be done, but there's no excuse for basically understanding this, because the questions, you can't just rote learn this. But the good news is once you do know it, I get very few of these wrong. My error rate on inheritance questions is much lower than it is on my error rate for much more the qualitative side of things, the ecology side of stuff, like, because I can work it out. It's more like mathematics. You can figure it out and you can be right. Whereas, yeah, so it's good. It's difficult to get your head around, but once you've got it, then you're there. Okay, so crossing over can only take place between homologous chromosomes, between the 2 pairs, the one you get from your mum and the one you get from your dad. So let's say it's chromosome 12. They line up next to each other during metaphase 1 of meiosis. This is, again, meiosis is super important. You have to understand how they move. And you can maybe then exchange some alleles. But if genes are not linked, then you've got one gene is on chromosome 12 and another gene is on chromosome 13 down here, then 13s are exchanging with 13s and 12s are exchanging with 12s, but you can't do crossing over between chromosome 12 and chromosome 13. That doesn't happen. So crossing over is really only to be considered if they are linked, because if they're linked, they're on the same chromosome, and therefore crossing over can shuffle things up a bit between those 2 genes if we're looking at the inheritance of those 2 genes. What is going on tomorrow? Let's have a quick look in the course. Gene pools and Hardy-Weinberg. So I'm going to probably chuck some Yeah, and then in the AS schedule, I'm doing some speciation, classification, phylogeny stuff. So I'm trying to link them together. Maybe I'm trying to be a bit cute with that. So they, in theory, the AS kind of, that's actually going to go down the pan when we get to gene technology, because there's not much precursors of that. I think I will, because the DNA, the simple DNA stuff that underpins all of that, I want to cover mostly the stuff the AS students haven't done yet. Okay, so yeah, that's what we're doing. Again, this is all based upon the feedback you gave me in that first session when you told me what you hadn't covered yet. Tisha, should you still be revising? You are going to be given a calculated grade. Because the exams are cancelled, anyone doing any kind of science-related degree will be expected to not just give up and pack up your tools, and that you will be expected to be of A-level standard when you get to university. So A, if your degree is going to involve any of this, then you need to make sure that you've covered everything and that you're up to scratch. Second of all, you're going to be given a grade. We don't know how that's going to be calculated. In general, on results day, 15 to 20% of students are not happy with their grade. In general, it doesn't matter whether that's when they've done the exams and the normal process has been followed through. There is going to be a solid proportion of you guys who feel that your grade is not just. Your only way of redressing that, of proving that you are better than this grade that you are not happy with, is by sitting an exam. And you are probably going to have about 5 to 6 weeks between them giving you that grade and you doing the exam. If you are worried about that grade not being the one that you want, and you want to do well in that exam, better than the grade that you're not happy with, then your only option is to keep things a bit fresh. Because I can tell you right now, after the summer holidays, if I go away for a month and I don't teach any biology and I come back and I'm like, I'm a bit rusty. It doesn't take long at all. Even for me, who's like, this is so drilled into me now. Like for you guys who have barely covered the content once, if you don't come back to it, you are not going to be able to excel in those exams. For me, it's also, if I was looking at it purely in terms of like, if I was coaching a student and you really, if your dream firm offer is a bit of a stretch, doing that exam is going to give you a very good chance of getting better grade than you would have otherwise done. Why? Because the grade boundaries are probably going to be low, because loads of people are not going to do any work or not do very much work. They are then going to try and sit the exam with 6 weeks revision, having done nothing for 3 months, and then they're going to do terribly, and the grade boundaries are probably going to be quite low. Also, you've got an extra few months to revise. So if you do want to apply yourself, you can totally boost your revision and sit that exam with an extra couple of months revision on top of what you would have otherwise done. And then you're going to be laughing because you're going to be better prepared than you were otherwise. Most students are going to be worse prepared. So you are actually going to be pushing yourself up the curve. So totally up to you. If you've been given an unconditional offer and you're not doing a science, and you're going to do media and film, and you did biology, chemistry and media, then maybe make sure your media is up to scratch. And you've been given an unconditional offer, it's your first choice uni. Sure, I probably wouldn't do mega amounts of biology and chemistry revision. But if that's not the case, then you want to make sure that you are. Also, you're going to get bored at some point in the summer. It's going to be a long, hard time, and keeping some structure, I think, is valuable. I'm going to go because Ronnie's going to be mad. No, Ronnie's in the chat. Yeah, I mean, I've been talking to them. They've not been concentrating. They've been on a bit of a break, but it is important you take your breaks. And on that note, I'm going to say goodbye, and I will see you all tomorrow. But I won't see you on Friday because I'll be on a plane. So I'll see you tomorrow, same time, same place. Spread the word. I want some more people joining these. And genuinely, if we don't get enough, then and we think we can do more useful things, we'll do something else. So tell your teachers, tell your classmates, get them to come along. Because if there's not hundreds of people joining us, then I think my time could be better spent helping more people in this situation in some other way. So yeah, spread the word. See you next time.